Hello and welcome everyone. Okay, so we are going to get started on Hematology Unit 2. We're going to follow up from talking about blood smears, making those and all that. So not only is it important for us to know how to make it, how to look at it, and what we're looking at, we now need to know what abnormalities we can see. Because it's great to see things that are normal, but are all of our patients normal? No, that's why they're patients. Okay, and that's why we're looking at their blood, because we want to know what's going on, what kind of disease, condition, or that sort of thing are we working with, and how, as we as veterinary technicians in the lab, can help with that. So it's important for us to understand morphological changes. I would also love if you all followed along with me in your book, this one you bought, page 63. We'll kind of piggyback off of making those blood smears in our hematology unit. So let's talk about some morphological changes. So in addition to us looking at a blood smear, counting the white blood cells, doing a platelet estimate, doing a differential, looking at all those features of the blood cells, we're also going to look at the abnormalities. What is wrong? What doesn't go here? And this is going to be the presence of any abnormal cell or potentially toxic changes, okay? Please note that yes, we're going to look and see if there's any abnormalities, but we also have to quantify. Now, don't get too stuck on the quantification. Just kind of add these in your notes. We will do this when we get into lab, like actual lab class, but there's going to be two methods used. So we could use a scale of like 1+, 2+, 3+, 4+, and that's going to be pretty relative. And then we could break that into potential percentages of like, if you're looking into that blood smear, and you see, let's say, 1 to 2, maybe 3 weird cells out of the whole thing that you're looking at, that whole monolayer. Well, maybe we put that as like a 1+, so maybe that's like a 5 or 10%. Okay. Again, don't get too bogged down. We're going to do this in class. Just make sure you have this in your notes so that way you know how to quantify. So 1 to 4+. We'll kind of look at that. If we were to look into a blood smear and we saw a bunch of, let's say, like hypersegmented neutrophils that could be potentially toxic, like a bunch, we would say, uh-oh, this is more than 75% of all the cells that are in here. This would be like a 4+. We got a problem. The other thing that we could do to measure this is do slight, moderate, or marked. Slight being like 10%, moderate being 25%, 50% being marked. What I'd like to do is I'd like for us to get into lab and kind of play around and see which one best fits. Okay. Again, these are relative, just like when we talked about with urine. You're looking at that quantification. It can get hard. It can get sticky. I love a good plus system, so my preferences are that 1 to 4, but we will get into lab and we'll see which one works best for you. Both are acceptable. Okay, our first abnormality that we are going to talk about is what is called nuclear hyposegmentation. So this is an early release of a banned neutrophil. Remember, we talked about banned neutrophils and how they're like a baby version of a neutrophil, because ultimately a segmented neutrophils should be segmented. And for a canine and feline, we're going to see about 3 to 5 different segments. For a horse, you could see like 7. So you got to know your species, got to know your history. Note that we talked about left shift last semester, and that left shift, if you remember, is the release of that immature neutrophil, but that's possibly due to a severe infection. So that's when an infection is so bad that it's wiped out all the neutrophils that the body could pump out, but then the body is pumping out underdeveloped or immature neutrophils to go assess the situation or the problem or the infection that's taking place. So left shift isn't a good thing because it's pumping out, again, neutrophils that aren't ready yet. They're not fully developed. So note that nuclear hyposegmentation is not a left shift. Okay, they look a little bit different. They look almost like a sleep mask at night, or like an 8, okay? Um, they're two different things, so they're under-segmented. So what is an example where we would see that? Well, I don't know if you remember from Diseases 1, but we talked about something called the Pelger-Hewitt anomaly. It's a congenital defect where we see hyposegmentation of all granulocyte nuclei. So do you remember which ones are granulocytes and non-granulocytes? Okay, so it can be any of the granulocyte categories. It's believed to be a result from an autosomal dominant trait. What is that saying? It's genetic. Okay, you've got your dominant and your recessive genes, so it's an anomaly of that dominant trait. It can cause skeletal abnormalities. It's seen at very, very young stages, oftentimes post-mortal, due to the fact that sometimes when they're that young, we can't catch it quick enough. And we're going to see this most commonly in Australian shepherds, okay? Side note, certain drugs can cause issues, can cause hyposegmentation of neutrophils, but it's not very common. It's just a side note, just so that you know. What are those drugs? It's not really sure, but medications can cause some issues with that. Okay, so then flip that and let's see nuclear hypersegmentation. So that's going to be the opposite. We're not low segmenting, we're hyper segmenting. And this is very common, yet still it's an abnormality. But why is it common? Okay, so this is going to be a nuclei with 5 or more lobes. Again, know your species, because if this is a horse, we're not really concerned. Dog, cat, what we got going on? Now note that if you just see with hypo and hypersegmentation, if you just see 1 or 2 of these, I wouldn't, I mean, you could write it down, document everything, but I wouldn't be like, oh my gosh, this dog has Pelger-Hewitt anomaly, or this animal has a problem. Not necessarily. It's when we see higher than most, like an abnormal amount. Because also keep in mind that is the way that we're storing the sample. How old is this blood? That's a big deal. If it's fresh, if it's new, if it's clinical, then we're going to notate that. Okay. Anyways, why are we going to see hypersegmentation? It can be due to the aging of neutrophils. Okay, so we know that the neutrophils have their life stages and have their process. Okay, so as it starts to go through its aging process, it may start to segment more because it's kind of that dying off stage, that apoptosis. The storage of this and the time, and then also it's common in poodles with macrocytosis, and that's going to be larger than normal cells. So is that an issue? Not necessarily. It's just something to know. Again, know your breeds and know your history. Okay, so with toxic changes, these are going to be seen on certain patients that have certain diseases or conditions where we see a change within the cytoplasm, remember the outer layer, and then we've got our fluid and everything inside that cell. That's where we're going to see the issues. So we have the fluid space, that cellular space within that cell. We've got some toxicity taking place. So again, this is commonly disease-induced changes, and we're going to talk about our neutrophils specifically. So these are going to be associated if a patient has a drug toxicity, if there is an infection, as well as if there's any inflammation taking place within the body. So for dogs, our canine patients, this is going to be more significant, and it's often suggested of a bacterial infection. Unlike in our cat patients, we can see these more commonly in cats, and they're not going to be clinical or severely ill, unlike we would see that with a dog. Cats are a little more hardcore, I guess. But we're always going to note anything in the medical record if we see anything. So what are some toxic changes that we can see? Well, one, we can see what's called a cytoplasmic basophilia. Okay, so basophils, those are going to be darker, right? We've talked about that before, and they're going to have like a darker nucleus than that of a neutrophil. And so they're overall just darker in color, unlike our neutrophils that could be kind of lighter, pinkisher, sometimes even grayisher. We then see dull bodies, which we'll talk about. We can also see vacuoles. It's kind of like bubbles or foaminess. Remember, vacuoles we can see in our macrophages or our monocytes, and that's normal. It's not normal to see those in a neutrophil, though. We can see what's called toxic granulation. This is rarer than the others, but it's still possible. So if you think about granulocytes and they already have granules in there, well, actually those granules can get a little teed off and they can be toxic. Okay, so that's toxic granulation. Don't get that confused with an eosinophil, though. We'll pop up some pictures here in a minute. We'll go through those. We can also see what's called like giantism, where they're just bigger than they should be. Note that this is going to be caused by a decreased length of time in the, like as the neutrophils will start to mature. And this is a process with the bone marrow. Basically, the bone marrow is making them too quick and pumping them out too quick. Okay, so they become toxic due to maybe infection or inflammation. And the bone marrow is just like, I'm trying, but I'm pumping them out and I'm making them toxic. Okay, so what are we looking at? Well, first we talked about cytoplasmic basophilia. Okay, so we're going to see one, that picture is not of this, so please hold on the picture. But it's going to be a streaky, diffuse, irregular blue appearance to the cytoplasm. It is due to the presence of polyribosomes and that rough endoplasmic reticulum, which you're like, excuse me, I don't remember chapter 1 from A&P. Well, let me remind you. Okay, so your endoplasmic reticulum has ribosomes, and those ribosomes are like your proteins and your amino acid building blocks, and they do a lot of fun stuff for protein synthesis and body function and all that. Okay, so it's ribosomal. We've also got like some DNA and RNA in there. So basically, it just means that we're going to see a darker than normal coloration of that cytoplasm, okay, because it's pumping out proteins and things that are going too fast. Remember, we're making it too fast. They're pumping them out too fast. So we can see a darker coloration, in short. So then we have this thing called dull bodies, and that is your picture, okay? This is the earliest and first indication of toxicity. We can see small gray-blue areas, and those are going to be ribosomes, and they're going to be seen in the cytoplasm and have some like immature toxic granulocytes. Okay, so we're going to see dull bodies. Those should not be there. Those are remnants of ribosomes. They were pumped out of the endoplasmic reticulum too fast, and they're not mature. They're toxic. However, we want to note that in healthy cats, we can have low numbers of these, and we want to notate it, but it's not like, oh my God, call the doctor. Emergency. We could see this as also like a storage artifact. And then sometimes even dull bodies in small numbers can just be for artifacts as well. Maybe your stain isn't clean and all that. So proper maintenance of your stain and making sure all that's good is important as well. Yeah, so those are dull bodies. Okay, now we have cytoplasmic vacuolation. This is going to be vacuoles in a neutrophil, in short, because we don't want to see those there. We see them in our macrophages and our monocytes. We don't want to see them in neutrophils. These are like bubbles, and they're kind of foamy, and they're not good. They kind of give that frothy appearance. And they're technically due to the degranulation of lysosomes, other kind of proteins helping things out in the cell. And if they're ever clear and not necessarily like foamy, it could be possibly due to a storage artifact. Also, it can occur within like 4 hours of your blood collection. Is it common? No. Like, is the storage issues common? No, because hopefully we're looking at our blood smear and looking at things before it's gotten to that point. Now, the question is that you're probably thinking, well, what happens if we draw blood and then we send it out? So like you have a patient come in for a wellness exam and you draw blood at 5 o'clock, but Antec or IDEX doesn't pick up the blood, which would be a separate lab entity where you send out blood work, doesn't pick it up until tomorrow morning. Is that going to be cause an issue? No, because hopefully you're storing it in the fridge, and hopefully you put the right amount of blood into the right amount of anticoagulant where you wouldn't see this issue. Again, it's not common, but that can happen. Now, what would the lab do? Well, if the lab saw this or there was an issue within the like the patient, hopefully they could run an additional test to rule some of this out. We then can see toxic granulation. These are distinct red granules in the cytoplasm due to primarily the granules taking up some stain. It's going to be rarely seen in our domestic animals, but we can more so see these when we get into our large animals, such as like horses, ruminants, and camelids. Make sure that we don't think that this is an eosinophil when we're actually looking at a neutrophil. Okay, let's look at some more pictures. Okay, try not to look at the note section and cheat. But number one, what do we see? Okay, it has the answer, but let's keep going. Number one, we can actually see this is cytoplasmic basophilia. We see that it's clumpy and it's blue. Now, what is another way that our cells could be altered due to us? Well, maybe we didn't stain it properly. Maybe we left our slide in the last purple stain for too long and we got distracted, or you had a seizure patient walk in, or we had to run up front for something, I don't know, in the hospital, and you left that in there, and then you're like, oops, left that in too long. Could we see stain issues, or it's going to be darker than it should be? Yes. So what should you do? Well, I hope you said you make another one, but if your patient's clinical and having issues, you should make another one. For the dull bodies, that's going to be number 2, and we can see these like blue chunky specks. For number 3, we can see cytoplasmic vacuolation. These are going to be, again, those foamy bubbles, and they're going to be inside the neutrophil, and they're not supposed to be. They're supposed to be macrophages or monocytes. Number 4, we can see the neutrophil is darker than the lymphocyte, which is not normal. And then additionally, number 5, we're also going to see those vacuoles. That's a pretty gnarly one. And number 6, it looks like an eosinophil, but it's not. It's actually toxic granulation, since this is a neutrophil. Again, we may be seeing some sort of infection or inflammation causing toxicity within that neutrophil. Okay, here are some more pictures that we could look at, and I'd like for you to write down what you think you can see. Will you see these pictures again? It has potential. It has potential. Maybe for like your quick quiz, so maybe you could put those in your notebook and write those down. Okay, so let's kind of switch gears and move away from like dull bodies and look at what's called inclusion bodies. So if you look in your book on page 64, figure 11.6, we can actually see, we see this picture right here, and it's a canine neutrophil that contains Ehrlichia. Okay, we can see Ehrlichia. The Ehrlichia part is noting that there's that inclusion body that's there. We can see them also in red blood cells, and this would be a possible canine distemper. So looking at a patient, if they had canine distemper and looking at a blood smear, we could see those inclusion bodies, and that would be a definitive, along with our clinical signs and history and signalment, that would tell us, okay, there's inclusion bodies here. This patient has distemper. We'll also go through more of these more so in class and look at pictures and differentiations. Histoplasma capsulatum, this will be a fungal example that we'll look at. So it's not necessarily like a virus. We can also see this in fungal as well. Mycobacterium, this is going to be a bacteria, and so on and so forth. Okay, so we can also see forms of atypical lymphocytes. One of those forms is an aerophilic granules. So we would be able to look at a lymphocyte and we're going to be seeing these like dots and granules that'll be kind of present. And we want to note that we can see this in canine ehrlichiosis, but we can also see this in normal bovine lymphocytes, like these kind of pink granulations that are taking place. And those are going to be from like an antigenic stimulation. And these atypical lymphocytes can also have basophilic cytoplasm, and they can kind of cleave onto the nuclei, where we don't have a lot of cytoplasm here, and it's kind of rounding and forming with that nucleus, and it's a little bit darker in color, so that can be seen. We can then see reactive lymphocytes. These are also known as immunocytes. They're going to have an increased basophilic or bluish cytoplasm. We can kind of see that there. This one's pretty dark. This one up in here. And they're also going to have more of an abundant cytoplasm of the cell. They're sometimes going to be larger and like have a convoluted or lobular shaped, not that, for a lymphocyte, not that perfect circle. And they're usually going to be caused by an antigenic stimulation secondary to a vaccination or an infection. So the body's like, whoa, 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 what is going on? And the lymphocytes are like the ones that are freaking out. So we can have the eosinophilic or the reactive lymphocytes. We can then have what's called lysosomal storage disorders. So you're like, wait, what's a lysosome again? Okay, so lysosomes, they're one of the organelles of the cell, and they basically are like the digestive system of the cell. They act as like a garbage disposal, and they help break down any like abnormal proteins. And those are going to be stored in the lysosomes. And so we can see this due to like the storage gone wrong, when there's like a disorder or a deficiency. It's a rare and inherited disease. And the substance, again, they're abnormally stored in the cells, which usually result in an intracellular enzymatic deficiency because they can't get rid of it. So most of the body cells are affected, but we can also see in monocytes, lymphocytes, or even neutrophils. When we see this, they could possibly be have vacuoles or contain some granules. Clinical signs that we would see are depending on the specific enzyme deficiency. Remember, those enzymes are absorbed into the body, then through those lysosomes, they process them, they digest them, metabolize it out, if you will, and so it depends on what's happening. But most of the time, it's going to involve skeletal abnormalities and maybe even some neurological disorders. Okay, we then have this thing in Berman cats where it's a neutrophil granulation anomaly. Okay, so neutrophils from the affected cats contain eosinophilic granulation, okay, and they're going to have like their fine point. They're going to be pretty small, and they're like almost magenta in coloration, as we can kind of maybe see from this picture. It is an inherited autosomal recessive trait, thinking back to like the dominant and recessive genes. And the neutrophil function is normal, and overall we can see this in like healthy cats where they're still going to be affected by it. Now, walk with me on this. We have to make sure that we differentiate between toxic granulation and mucopolysaccharidosis, where it's the body can't break down long-chain sugars, leading to lysosomal, there those are again, storage disorder. And we have to make sure we have to differentiate from that and GM2, which is gangliosidosis, which are issues in fat breakdown. Okay, so again, this is another type of that lysosomal storage disorder. So Berman cats, they can have this weird neutrophil anomaly where they have like this pink granulation, but it's not an eosinophil. Okay, and it can happen also in healthy cats, and sometimes they can have clinical weird signs, but we can see this. Okay, so the Berman. We then, you should remember this, hopefully, from diseases one where we talked about Shediac-Higashi syndrome. It's an inherited disorder that we see in cats, and the neutrophils have large fused lysosomes within the cytoplasm. We look at approximately 1 in 3 or 4 neutrophils will contain those fused lysosomes. The stain will be kind of a light pinkish color, and it's almost like eosinophilic, but they're bigger than those granules of an eosinophilic granule. And they have this, as we can see in this picture, they have that neutrophil form. The neutrophil function is going to be abnormal, but affected animals are generally healthy. Okay, so the neutrophils may be doing some weirdness, but clinically, your patient may not be severely affected. Although affected animals may bleed due to abnormal platelet function. So most often, we're going to see this in our Persian cats, like we've talked about before. But we can see Shidaya-Higashi syndrome in cattle, foxes, and maybe some other kind of wildlife and that sort of thing. So make sure that you're able to have that picture like in your notebook too, and differentiate between our lysosomal storage, Berman, and then your eosinophils. We then have this siderotic granules. We got a lot of granulation happening. These are going to be granules of the hemosiderin, and they may be present in neutrophils as well as monocytes of the animals with hemolytic anemia. And why is that? Well, when red blood cells break down, they're breaking down hemoglobin too. And hemoglobin has that heme color, that heme pigment, as like its leftovers. Remember, hemoglobin is also that storage form of iron, and so we're going to have that leftover. Okay, it's a leftover of red blood cell breakdown. That oxygen carrying component of hemoglobin breaks down, and it has like that pigment color, like we talked about in diseases 1. Okay, so how do we tell the difference between these? Well, here in this picture, you can see that there's some granulation, or I don't want to say granulation, there's pigmentation that's occurring within these cells, okay? Specifically these red blood cells. The way that you differentiate this is the stain that you use. So if you were to use DivQuik stain, like we use for ear cytology, or just in general to stain our blood smears, you are going to be able to see dull bodies, but you would not be able to see siderotic granules, okay? The siderotic granules have to have the Persian blue stain to be able to look at that, okay? We then have this thing called smudge cells, also known as basket cells. I've never called it a basket cell, mostly just smudge, because that's a fun word too. They are degenerative leukocytes that have ruptured. So they are starting to degenerate and then just go, okay? They're not significant unless they're in large numbers. Your VTD loves to ask this question, so I definitely make sure you remember this one. Large numbers are going to be associated with leukemia, okay? Big star here. Big star here. Small numbers can be produced as an artifact during your smear technique. So how you're forming that smear, like your shwee, okay, that's really important. How we're taking care of the blood, how our slide is going through and all that, that's important because if we see a lot of smudge cells, do we have leukemia or did you just get wonky with your smear? Again, in large numbers, you would, well, let's just redo the slide. Let's just redo the slide. Maybe need another blood sample because that would be not a great diagnosis, and we want to make sure that that wasn't an error on our part. Okay, just as a little refresher from Diseases 1, we talked about nuclear changes. We have pycnosis, karyarexis, and karyolysis. So pycnosis is the condensing of the nucleus as the cell dies, and that's going to be more of like a rapid cell death. It's like being picked apart. Karyarexis is the fragmentation of the nucleus after cell death. And we can see, so pycnosis is kind of here. It's like picking little picky pieces apart. And the karyarexis is going to be that fragmentation. So it's like broken up into different fragments. And the karyolysis is going to be here, which is the degenerative change to the nucleus by the dissolution of the nuclear membrane, and it just kind of fades away. And that's that cellular aging process. They're usually going to affect neutrophils as it's associated with the presence of like sepsis or like that severe infection leading to a septic patient and that sort of thing, because the neutrophils come in, they do their job, and then maybe they get wiped out. Okay, so make sure that you remember your nuclear changes. Okay, we've had so much fun with white blood cells. Now we are going to move into red blood cell morphology. We'll cover a lot of this. I really highly suggest that you put many, if not all, pictures and that sort of thing for your morphology. Not only will you need this for this class, but you're going to get out in the field and you're going to be doing blood smears and needing to be looking at morphology and all that. So let's get started on some red blood cells. Okay, it's important for us to understand the variations in cell arrangement. Okay, so there's something called a rouleau, which is the grouping of red blood cells in stacks. Okay, these pictures are an example of that rouleau. They kind of look like stacked coins, like if you were to take like a roll of coins and just like kind of lay them out, it'd be like rouleau. This is normal in horses, and it may be present in cats and pigs. Increased rouleau formation is seen with increased fibrinogen or globulin concentrations. It can be an artifact of prolonged storage or refrigeration, which we might have seen in our blood samples as, you know, we're having to save some things for a while. Okay, so if you have to do a blood smear, and you look and you find this rouleau, if it's a dog, something's weird. Are we agglutinating? If it's a cat, oh, well, this is a normal finding that we'll see. Speaking of agglutination, there's variations in that cell arrangement as well. We can see agglutination. This must be distinguished from rouleau. Okay, so agglutination is like a glob, okay? Not the like stacked coins, where it's kind of like a smooth stack, okay? Agglutination is like the clumped together, like a mold. Autoagglutination is going to occur in an immune-mediated disorders, such as red blood cells are going to be coated with antibodies, resulting in like that bridges in those clumps. And an example of that autoagglutination is we can see IMHA, so immune-mediated hemolytic anemia. We can see this agglutination taking place, okay? Again, it's different from rouleau. What we'll do in class is we will take a drop of blood and a drop of saline, and we'll kind of look at it all. And here is an example of the test that we could do. It's not that fancy. It really is a drop of red blood cell and a drop of saline, and you look to see, is it smooth, like a red liquid, like it all comes together, or did it disperse and kind of become this like speckled egg, like dee, or is it like smooth and everything's colored in? Okay, so agglutination, we don't want to see that. Okay, we don't want to see that because that tells us, what is the body doing? What are our red blood cells doing? Okay, it's different from rouleau. So agglutination and rouleau. Okay, so anisocytosis is a variation in red blood cell size. We can see these in macrocytes, microcytes, or even both. So macrocytes are going to be larger than normal with increased MCV. We usually see this in young polychromatic erythrocytes. We can see this with reticulocytes for sure. And the microcytes are smaller than normal. There's a decreased MCV on that CBC, and we can see this potentially with some iron deficiency, okay? It's common to find this in normal bovine blood, but anisocytosis, a variation in red blood cell size. The morphology of your red blood cell will affect the cell size, right? So if you have, which we'll later talk about some other ones like echonocytes or schistocytes, they're going to have different size, right? But overall, the variation should be similar with each other. So if we look at these pictures, we can see that a lot of these red blood cells, there's some small ones, there's some big ones. We got something kind of going on. We need to kind of assess what is happening here. If we see this in like young animals, we could possibly relate it to developmental stages, as we know that like puppies and kittens can have slight anemia due to the bone marrow just trying to kick in and learn to pump things out as it should and develop. But overall, having those variations is not something we want to see. It could also indicate some sort of like blood disorder, which we'll talk about that a little bit later. We then can see variations in cell color. So the term polychromasia refers to the erythrocytes that exhibit like a blush tint when stained with the Romanowski type stain, or also known as Wright stain. So figure 1115, we can see that there's this blue tint, and it's a result of the presence of the organelles in the cell that remain within that cytoplasm. Okay, so therefore, these are going to be young cells, and when stained with pneumethylene blue, these cells will appear as kind of like reticulocytes, which we'll talk about a little bit later. But polychromasia is the variation in cell color. We then can see hypochromasia, which are red blood cells with a decreased staining intensity. This can be from like insufficient hemoglobin within the cell, so maybe we're anemic, maybe we've got an iron deficiency. We can also have like a pale central region with like dark stained periphery. Again, iron deficiency can cause it. We have to distinguish between anucleocytes and then taurocytes. We'll talk more about that later. But true hypochromasia almost always will have microcytes and will have that decreased MCB. We then see normal chromasia, which is normal staining intensity, which is what we want, and hopefully we get. Don't leave it in the purple for forever. Okay, we now can see on the opposite side, hyperchromatophilic cells. These are going to be more darkly stained red blood cells. They give this appearance of like too much hemoglobin, so too much iron, that sort of thing. But a cell has a fixed capacity for hemoglobin, so oversaturation really can't actually occur. And we'll see these maybe in some microcytes and maybe some of those spherocytes that we look at. Spherocytes are going to be another type of red blood cell that we're going to talk about and look at too. Okay, next we have poikilocytosis. These are going to be cells that are abnormally shaped. It's really not actually that helpful in diagnosing things. But we're only going to use this when morphologic abnormalities cannot be described more specifically. So if you saw like kind of these guys, well, maybe things got smushed as you were making the slide, and so we could be like, look at that poikilin around, some poikilocytosis. It's also a fun word. And then we can see what's called schistocytes. So schistocytes look crappy. They are red blood cell fragments, and they're like a deflated balloon, like, look, okay? They result from intravascular trauma, possibly due to the shearing of the red blood cell. These aren't good. Schistocytes are not good. They may be observed with DIC, or disseminated intravascular coagulopathy, or we could have potentially hemangiosarcoma, so that would be a type of vascular neoplasia or cancer. So schistocytes are not something you want to see. If you had a patient that presented with DIC, you did a blood smear, you will probably find these because we have intravascular red blood cell trauma occurring. Okay, we can also see acanthocytes. These are also known as spur cells, like maybe like the spur of a boot. Is that a thing? Well, whatever. They can be irregular and like kind of have these spine-like projections, and they're seen in patients after altered lipid metabolism. So what are some examples of that? Well, we can see them in cats with hepatic lipidosis. So hepatic in the liver. So we've got some liver deficiencies. We've got some liver disorders. With hepatic lipidosis in particularly, remember cats have a hard time with, they're obligate carnivores, so they have a hard time with digestion if they don't have a proper amount of fat and protein. So their liver can basically kind of freak out. So we can see specific cells if a patient with hepatic lipidosis arrived. Dogs, we can see this with hemangiosarcoma of the liver. So if the liver has cancer, those cells are affected, they're upset. We can also see this especially with middle to old age large dog breeds with concurrent regenerative anemia. So that's acanthocytes. Echonocytes are known as Burr, not spur, Burr cells, and they're going to have spine-like projections as well, but they're going to be more uniformed and evenly kind of spaced out. It's a common term for this abnormality in crenated, is crenation. Remember we had those red blood cells in the urine and it was crenated. But we term this as echonocytes. Okay, it's going to be different. They can be artifactual due to slow drying of your blood smear, prolonged storage, or underfilled EDTA. So the tubes, when they say fill in half a mil, 0.75, 1 ml, you definitely put in the correct amount because that could affect your blood and your cells. We see this in dogs with renal disease, lymphosarcoma, and snake bites. We can definitely see this in snake bites. I have several times had patients that presented for a possible snake bite, and then we looked under the microscope to help in our definitive diagnosis, and we were able to see echonocytes, CV cells present, which is always really fun to also show the owner or the client or customer, wherever you work, and we can be like, hey, yep, you cat or dog got bit by a snake for sure. And also use context clues and clinicals and histories to get your full diagnosis, but that is a way. We see this in horses after exercise, and we can also see some echonocytes present in healthy pigs, but this would be a normal thing. We can see drepanocytes. These are also known as sickle cytes. They're normal in deer and Angora goats. Kind of pretty and wispy. We can also see keratocytes, also known as helmet cells, also known as blister cells, also known as bite cells. Why? I don't know. Shape, morphology, maybe. Keratocytes, I don't really care what you put on the exam. Just know what you're looking at, okay? This presence is associated with hemangiosarcoma, neoplasia, glomerular nephritis, so issues with our kidney, and also we could have issues within the liver. Keratocytes. We then can see spherocytes. Spherocytes are going to be small, darkly stained red blood cells with reduced or no central pallor. Okay, they have reduced amount of membrane surface area due to partial phagocytosis by those macrophages. Remember that Pac-Man? It's in response to the presence of an antibody on the surface of that red blood cell. Again, trying to do the job. We can see a normal volume, but we can also see a decreased MCV. They're hard to detect in species other than dogs, really. It can be suggestive of immune-mediated hemolytic anemia or IMHA, but you'd want to look at other factors too. What's that other test that we can do to look at IMHA that we talked about? Slight agglutination. Okay, we can do that agglutination test. And for the spherocytes, they may be seen following transfusions, snake envenomation, red blood cell parasites, and also we can see this in zinc toxicity. Okay, we can see leptocytes. These are cells characterized by an increased membrane surface, and they're relative to the cell volume. We can see target cells, also known as podocytes. These are leptocytes with central area of pigmentation surrounded by a clear area. Then there's like a dense ring of the cytoplasm. So these target cells are pretty much going to be like a target symbol, okay? They're going to have, you may see like few to normal, and they can be seen with patients with anemia, liver disease, or anything that's possibly genetic or inherited. Okay, so we see these target cells. They basically make like a target, okay? Stomatocytes are leptocytes that appear as folded cells. So they have like this slit-like pale region of the center of the cell. So here's our stomatocytes. They can be artifacts if the red blood cells are kind of like folded of the areas of the pallor, or if they're perpendicular or have that feathered edge. Kinesicytes, also known as bar cells. This is a type of leptocyte with a bar of hemoglobin kind of across the center of it. So one thing that I forgot to mention with your stomatocytes, if you go to the, in lab we talked about that additional resource for the eClinPath. If you check that out, you'll have not more of a breakdown, but a little bit more of a breakdown of these types of cells and what we can see. So like, for example, stomatocytes, I didn't put on there what we can see. So here's what we can see. Disorders could be hereditary stomatocytosis in dogs. We see this in like Alaskan Malamutes and miniature schnauzers, as well as a peekapoo and Pomeranians. We can see those stomatocytes. And we could also, physiologically, we'll see those in woodchucks, manatees, and dolphins. Good to know. Target cells that we talked about, we can see these with iron deficiency, alterations in lipid content, and those liver disorders. So that's the one that's right under it. So that website's also a wonderful resource for you. We have elliptocytes or ovalocytes. These are normal in camelids and non-mammalian animals. In other species, they can be associated with lymphoblastic leukemia, hepatic lipidosis, portosystemic shunts, and also glomerular nephritis. So we've got some kidney and some liver issues taking place. Eccentrocytes, we can see hemoglobin pushed to one side. Okay, and with that, we can see that in patients with DKA, neoplasia, Babesia canis, ingestion of garlic, onions, and acetaminophen. So garlic and onions can cause anemia. We can see these because there's some weird red blood cell interaction going on, and the hemoglobin's like whoop to one side. Dicryocytes, they're going to be teardrop shaped. They're seen in myelofibrosis or fibrosis and myeloproliferative diseases. We can see these in llamas and alpacas with also associated iron deficiency. Though keep in mind, dicryocytes might not be anything to decry about because it could be an artifact. It can be identified as an artifact if the tails all pointing are all pointing in the same direction. So dicryocytes, they're bacterial type, don't decry about it. So here's just a chart and an illustration of the red blood cells, some that we've talked about. This is a little more comprehensive than I'm going to fully expect you to know. So if we haven't talked about it or we're not about to talk about it, you don't necessarily have to worry about it. However, it's a fun picture and it's a good little stopping point for us to like check, kind of review. You may see it in bingo form one day. Okay, just like we talked about with white blood cells, we can see inclusions in red blood cells. First one we'll talk about is basophilic stippling or stippling. This is going to be the presence of small dark blue bodies within a red blood cell, like in this picture. They can be observed in right stain cells and the presence of residual RNA. It is common in immature red blood cells of ruminants. Occasionally, we can see it in cats responding to some form of anemia, and we can see this in patients that might have lead poisoning. So if you see this speckled egg, that's basophilic stippling or stippling, and raise your hand. And also let a doctor know what's going on with this. We then can see howl jollies. These are basophilic nuclear remnants in young red blood cells in response to anemia. The nuclear remnants will usually be removed when it's passed through the spleen, but increased numbers are going to be seen in splenic disorders or after a splenectomy. Okay, so if the spleen is going to swoop those remnants out, well, maybe it's not going to be there and able to do that. So howl jollies. Think of like a jolly berry of Christmas. Okay, let's look at Heinz bodies. Heinz bodies are going to be round blue structures representing denatured hemoglobin, which becomes attached to the cell membrane. So these are examples. Make sure you know the difference between howl jolly and these Heinz bodies. They're going to appear as like pale area with the right stain, and they're going to be blue with new methylene blue. Now, we can see those with right stain as well as in our diff quick. They're just kind of going to look like this. We can see it's normal to see Heinz bodies in cats in like 5% of the red blood cells. So if you see like more than 5% or an abundance of those, we might need to assess that because increased amounts are going to indicate lymphosarcoma, hyperthyroidism, and potentially even diabetes. Okay, so these are Heinz bodies. Okay, so you all know that red blood cells should not have a nucleus. But what does white blood cells? Okay, good job. If a red blood cell has a nucleus or a big dot in there, we would actually note this as a nucleated red blood cell. And this means that there is an early release of the immature red blood cell during like anemia. But occasionally it's observed in like non-anemic animals. So if you saw like a nucleated red blood cell, maybe one in the entire slide, are you going to write home about it? No, I mean, notate it. There's nothing wrong with documentation. Made that up. All red blood cells of non-mammalians are going to be nucleated. Okay, so if we look at this picture, this is going to be avian or reptile red blood cells. They have a nucleus. Cats and dogs should not have a nucleus. If they do, then it's a nucleated red blood cell, and that means the bone marrow pumped it out before it was mature. Like, wait, you're not ready, but I want to drive the car. Okay, and now you're still nucleated. You weren't ready. If you have a patient that is anemic, okay, and you see nucleated red blood cells, this does tell you, even though you don't want to see it, it does tell you that the body is trying, okay? They're trying to produce, and the bone marrow is trying to kick in to release these, okay? They're not fully developed, but the bone marrow is trying. So you may see this after like a transfusion. We can see this reported in a differential cell count as nucleated red blood cells per 100 white blood cells, and it's like incorporated into the following like equation for these. We'll do this kind of in lab, but here's that equation as that breaks down. Again, here's a nucleated red blood cell. Okay, so we don't want to see those. However, we can see those in, like, if you saw a couple, it's not going to be terrible, but again, look at your patient. Are they anemic? Did they just have a blood transfusion? If we're seeing them for a patient that's anemic or IMHA, it's like, oh, well, maybe the body's trying. Parasites, my favorite. They can be confused with drying artifacts and stain precipitation. Okay, drying artifacts can be like refractile, so we have to know what we're looking at. History helps too. Most common blood parasite in small animals is going to be Ehrlichia as well as Mycoplasma. Okay, Mycoplasma haemophilus or feline red blood cell parasite, also known as Hemabartonellus or feline infectious anemia. we can see that. They're going to be small, like coxoid, like coxi, like round, or they can also have like a rod shape or ring-like appearance, and they're going to have those dark, like stained little dots, basically. We can see it on the periphery of the red blood cell. So it's not going to be inside like our Heinz bodies. It's going to be on the outside, okay? It's going to be on the outside, the periphery of that red blood cell. It is a cytosolic parasitism, so the blood should be examined several times of the day, before, right after. We should evaluate that blood. Now, is this something that you're going to potentially see? Maybe. I mean, we saw a cast on day 1, so in urine. So is this something that you're going to necessarily see? Maybe, maybe not, but it might be something that you have to send off to the pathologist for them to review. But make sure you know the difference between the Heinz bodies, where it's inside, and then these are on the periphery. Okay, so with our Ehrlichia, we can see this in monocytes or neutrophils. We can also see some anaplasma species. So let's take a look at these on our next slide. As we know, these can be like tick-borne diseases, infectious bacterial organisms, maybe some rickettsia, Ixodes scapularis from deer ticks, so on and so forth. So let's take a look at those on the next slide. Okay, just to kind of break these up into a little bit more, if we look at this picture down here, we can actually see that these are platelets. So we can see Merlot, that like kind of pigmentation. We can actually see this in some platelets, and this would be an example of anaplasma platus. It kind of helps with the wording. Okay, so we can see this type of anaplasma. Ehrlichia, here's going to be that Merlot granulation as well, that Merlot. Here we are again within those. We then can see the form of anaplasma marginale. We're going to see those kind of more on that periphery as well. And then here is, we're going to talk about on the next slide, this is going to be our epierythrozoa that we'll see. So with epierythrozoa, we can actually see this, we can see this in swine, in cattle, as well as llamas. This we would see, like, for example, with patients with mycoplasm. We're going to see like those, they're kind of like cocci and sometimes even rod-shaped, maybe even ring-shaped, and they're going to have kind of that appearance. So that's going to be another type of parasite. I'm pretty sure you've talked about it before, the cyticozoon or cyticozoan felis. This is where we're going to have that bejeweled effect in our red blood cell. They're going to be shiny, kind of jewel-like. This is going to be, your book says that it is a rare cause of hemolytic anemia in cats. It's becoming a little more common that we can see. Unfortunately, if we see this, this means that that patient does have a more guarded prognosis. And then Babesia, if you see it in cattle, that's reportable. You should probably say something. Not probably, you should. Babesia, we can see this cattle, horses, dogs, their own type. But here we can see they're usually grouped together, like 2s or 4s, 6s. And so we can see that parasite as well. Man, we did it. Check out that morphology. I can't wait for us to get into lab class and for us to like kind of look at these things. Hopefully we won't see some of the like parasites or abnormalities, but we will do flashcards and we'll talk about, you know, how they correlate and what does that actually mean. So quantifying those morphologic changes are important. We'll do that. Looking at those abnormalities, inclusions, parasites, and all that. We'll do more of that when we meet again on Thursday. Make sure that you've taken notes. Make sure that you've looked at your notebook and have good descriptions along with pictures in those, not only for your quick quiz in this class, but also for your life. Okay, great. I hope you learned something and have a great day.